Hello everyone. Today's topic is glycolysis and TCA cycle, which are the most important metabolic pathways by which our cell produces energy. Our cell uses the food to metabolize to produce the energy. The mitochondria is the main site for metabolism. It is also called as the powerhouse of our cell. Why it is called? Because it produces the ATP molecules, which are the energy coins for cell. In this topic, we will discuss the process of glycolysis and TCA cycle, as well as we will go through the structure of mitochondria and ATP synthesis and their working. The metabolism of food is called as cellular respiration. We intake food and oxygen which get metabolized through the mitochondria. The process of glycolysis takes place in the cytoplasm, whereas the Kreb cycle takes place in the mitochondrial matrix. And there is electron transport chain present in the mitochondrial membrane which convert the NADH molecules into ATP, and thus our whole motive is to production of ATP through the metabolism of food. When we see the structure of mitochondria, it is a double membranous structure. It has its own DNA and ribosome, so it can produce its own protein, and it can replicate itself. Its inner membrane has various folds called cristae, which has a very important factor called as F0F1 complexes, which forms the ATP synthase enzyme. The base of ATP synthase is of F0 base. It acts as an proton pump at the time of ATP synthesis, whereas it had termed as F1, which stores the energy in the form of ATP. This is how the mitochondria and ATP synthesis works. ATP is termed as adenosine 5 triphosphate. It has a ribose sugar which is attached to the adenine nitrogenous base. You can see the structure of adenine and ribose sugar. But the most important thing is this triphosphate bonds which stores the energy. When ATP molecule detaches the phosphate group, it releases the energy, and this free energy is used by our cells, and it is converted into adenosine diphosphate. And similarly, when energy is produced, it restores the energy in the form of additional phosphate bond and converted into ATP. Thus the ATP and ADP cycle restore and reabsorb the energy produced by cells. When we see the metabolism, our food we take contains carbohydrates, fats, and proteins, which are converted after the digestion into elementary molecules such as sugars, glycerol, fatty acids, and amino acids. All these elementary molecule goes through the glycolysis and citric acid cycle, which then finally converted into ATP. The NADH are produced in this process, which go through the oxidative phosphorylation in the electron transport chain of mitochondrial membrane, which is then converted into ATP molecule. So our whole motive is to produce the ATP molecules through the metabolism of food elements. The process of glycolysis is the conversion of glucose molecule into pyruvate molecule. Glucose is a 6 carbon molecule. Firstly, it reacts in the presence of hexokinase and use the energy in the form of ATP. Always remember, whenever the kinase enzyme present, it converts the ATP into ADP and vice versa. So it has an important role in the ADP-ATP cycle. The kinase enzyme always related with ATP production or its consumption. So glucose in the presence of hexokinase is converted into glucose 6-phosphate. In the presence of isomerase enzyme, it is converted into fructose 6-phosphate. Then again, phosphofructokinase use the ATP molecule to convert it into fructose 1,6-biphosphate. Then two phosphate groups are attached. Then in the presence of aldolase, it is converted into two molecules of glyceraldehyde. You can see here the ring is breaking down, and three carbon atom of glyceraldehyde is produced. Then in the presence of dehydrogenase, it produces 2 NADH. Always remember, whenever the dehydrogenase enzyme is present, it produces the NADH. So this is how the NADH molecule is being produced here. Then again, in the presence of phosphoglycerate kinase, two ATP molecules are being produced here. Then 3-phosphoglycerate is produced, which is with the help of mutase enzyme is converted into 2-phosphoglycerate enzymes. Then the enolase enzyme release the two water molecules and convert it into phosphoenol pyruvate. Again, the pyruvate kinase enzyme produce the two ATP molecules and finally convert it into two molecules of pyruvate, which is a 3-carbon containing molecule. So, if we summarize the glycolysis, we have consumed two ATP molecules at the starting point, and we have gained a total of 4 ATP molecules. So we have a net gain of 2 ATP molecules through the glycolysis, as well as we have two molecules of NADH which will go through the electron transport chain and it will give us ATP, as well as we have gained two water molecules which will be used in Kreb cycle, and as a substrate we get two molecules of pyruvate. This pyruvate, which is being produced in the glycolysis process, is go through the link reaction in which dehydrogenase enzyme will convert coenzyme A to the pyruvate and produce acetyl coenzyme A, which will be used in the Kreb cycle. So this is how the process of glycolysis and Krebs cycle are interrelated to each other. It produces NADH because dehydrogenase enzyme is used. So one molecule of NADH is produced, which will go through the electron transport chain. This is the TCA cycle. It is also called as Citric Acid Cycle and Krebs Cycle. It has various names. So oxaloacetate firstly react with water molecule in the presence of citrate synthetase and convert the acetyl coenzyme A into citrate molecule. In the presence of aconitase, it release the water molecule and convert it into cis-aconitase. Again, the aconitase enzyme use this water molecule and convert the into isocitrate. This then isocitrate dehydrogenase enzyme produce the NADH and CO2. We have seen there, whenever the dehydrogenase enzyme is present, the production of NADH takes place. So it is then converted into alpha glutarate. Alpha glutarate dehydrogenase enzyme again produce the NADH molecule and carbon dioxide and produce the 4-carbon atom molecule succinyl coenzyme A. Then in the presence of succinyl coenzyme A synthetase, one molecule of GTP is produced, which will also go through electron transport and converted into ATP. And this is how succinate is produced. Then this succinate, with the help of succinate dehydrogenase, release the FADH2 and convert it into fumarate. Then in the presence of fumarase enzyme, with the presence of water molecule. It is converted into L-malate, then malate dehydrogenase enzyme again produce NADH and converted into oxaloacetate. So this is how this cycle runs throughout the process, and it releases total of 3 NADH molecules. One pyruvate, or we can say one acetyl coenzyme A, produce 3 NADH, one molecule of FADH2, and one molecule of GTP. But we have seen through the one molecule of glucose, the glycolysis process gives us 2 pyruvate. So we will have 2 acetyl coenzyme A. So all these yields will be doubled. So we will have a total of 6 NADH, 2 FADH2 and 2 molecules of GTP as the end product of TCA cycle. These all energy carriers will go through the electron transport chain. So this is how the TCA cycle produces energy carrier molecules. When we summarize the process of glycolysis and citric acid cycle, we have seen the glycolysis has converted glucose into 2 molecules of pyruvate producing 2 ATP, and it has produced 2 NADH, then which by for producing acetyl coenzyme A. Then the pyruvate oxidation also produces 2 NADH in the link reaction. Then it goes to the Kreb cycle in the mitochondrial matrix where it produces 6 NADH and FADH2. All these NADH and FADH will go to oxidative phosphorylation, which will ultimately produce 28 ATPs. So this is how a complete metabolism of one molecule of glucose will gives us 32 ATP through their metabolism. So a complete of glycolysis plus citric acid cycle will gives us 32 ATPs. This is how the metabolism of foods takes place. I hope you have understood a brief of glycolysis and TCA cycle. Thank you very much.